putting together this meeting and making sure that it goes forward despite the pandemic, and also for inviting me. Also, I'd like to say hi to all the attendees, and I hope that you're all staying healthy and safe from the coronavirus. So as Arvind mentioned, I'm going to talk about a topic that is really dear to my heart, and it's post-transcriptional programs of cancer. So I will start by a brief introduction about the research that we do in my lab. So my lab is a computational biology group at McGill University, and we study gene regulation using computational approaches and the connection between gene regulation and human diseases. As you know, gene regulation encompasses a lot of different steps. It starts actually from the DNA with DNA rearrangement and copy number variations, and then RNA transcription, processing of the RNA, export of the RNA, decay and stability of the RNA, RNA translation, post-translational modification of proteins, all these different steps. And what we are interested in my lab is these two aspects of gene regulation: transcriptional regulation and post-transcriptional regulation of gene expression, and their connection to human diseases, but specifically to classes of human disorders, cancer and neurodegenerative disorders. So the main questions that we'd like to answer in our research are basically these ones. So from the perspective of just characterizing gene regulation, I think one of the first things that we'd like to do is actually to measure the phenomenon that we want to study. So we are basically interested in knowing how we can measure the rate of transcription and the kinetics of post-transcriptional gene regulation from transcriptome profiling data, which is one of the topics I will talk to you today. And then we also want to know what are the factors that determine these kinetics. So when we talk about transcription, for example, how do transcription factors interact with the genes? What is their function? How do they regulate the transcription of any gene that they bind to? And from the post-transcriptional regulation perspective, what are the factors that bind to the mRNA and regulate the processing and the life cycle of the RNA? And then we'd like to see how these things are connected to human diseases. So basically, again, talking about measurements, can we measure these kinetics in the context of human disease and understand what are their differences between the disease state of the tissue and healthy normal tissue? And also, what are the factors that derive these differences? So basically a network-based approach where we look at the regulators of these events across disorders, and we try to understand what is the role of these networks and these programs in development of these different disorders or their progression. So the research in my lab, obviously like any other lab, is driven by these fantastic scientists, the students and postdocs in my lab. And I thought that since I'm going to talk a lot about a network-based approach, maybe this kind of network-based representation of who is doing what would be fun to look at. I really had a lot of fun putting together this slide. So every one of these is a great scientist with their own story that I think merits its own talk, and that makes it a little bit difficult for me to decide what to talk about. But I decided to talk about this part of our research, the post-transcriptional regulation of genes, and how that concept connects with cancer. And there is a reason that I decided to talk about this. The main reason is that this is really a very not well studied aspect of gene regulation. So as we heard in some of the talks this morning, transcriptional regulation, which is the part that is here now, it's missing from the slide, that is the part that a lot of people have focused on, and there's a reason for that. It's the first step in gene regulation, is very important, but then that has led to kind of a neglect over post-transcriptional gene regulation. And as I will try to show to you today, there is a lot of interesting stuff going on. from the perspective of post-transcriptional regulation in cancer. So this is one reason I decided to talk to you about post-transcriptional regulation today, the fact that it's not well studied. And the other reason is that it's, as I mentioned, really dear to my heart because it kind of represents my journey through research from my undergraduate studies to where I am right now. So basically, I started looking at post-transcriptional regulation when I was an undergraduate student at the University of Tehran. And actually, at that time, I had no idea that what I'm looking at is post-transcriptional regulation. You know, I was basically fascinated with how certain genes select certain codons to encode their amino acids. So some genes prefer a specific set of codons, some genes prefer another set of codons. And I was looking at this kind of bias in codon usage in prokaryotic organisms in bacteria, particularly E. coli. And basically what I figured out is that, okay, we can actually predict the expression of a gene in E. coli to a good extent, not completely, but to a good extent, just by looking at their codons and what kind of codons they prefer. And now I know that this actually, to some extent, connects to post-transcriptional gene regulation. It's its own fascinating story. A lot of studies have come out showing how codon usage affects the gene expression, affects the stability of RNA, translation of RNA. So before actually I knew it, I was really interested in post-transcriptional regulation. But then in 2007, I traveled 10,000 kilometers to Montreal, to McGill University for my PhD studies. And during my PhD, I studied this organism, this unicellular eukaryotic organism called Trypanosome brucei. It's a human parasite, but it's really a fascinating organism because even though it's eukaryotic, transcriptional regulation plays very little role in regulation of gene expression in this organism. So basically, the genes in this organism are transcribed as these giant polycistronic mRNA. So this is a eukaryote, but it has these polycistronic mRNA, and then these mRNAs are kind of spliced into individual genes that encode different proteins, and then they are regulated post-transcriptionally. So everything happens post-transcriptionally. That's why it's a really good model for studying post-transcriptional gene regulation. At the time, I was looking at the effect of different elements within the RNA on post-transcriptional regulation of genes in this organism, what kind of RNA binding proteins recognize this. And then in 2012, I took a relatively shorter trip to Toronto, so not as far as my other trip, where I joined University of Toronto as a postdoc. And there I was, one of the first things that I started to look at was post-transcriptional regulation in the human genome and the role of RNA binding proteins that bind to the mRNA sequence, their conservation, their function in post-transcriptional regulation. And then in 2016, my journey kind of came back to Montreal, where I joined McGill as an assistant professor. And this is where the rest of the story that I'm going to talk to you today actually happens. So the first part of what I'd like to talk about is about measuring RNA stability from Rna-Seq data. So as I mentioned, I think one of the first steps of understanding anything is actually measuring that. So you know, this is the life cycle of the RNA, right? So the RNA gets transcribed from DNA into this premature, unspliced, unprocessed RNA, but then it goes through splicing and RNA processing. Introns are spliced out, other things happen, and then we have the mature RNA. And then at the end, this mature RNA gets degraded and is recycled, basically, the building blocks, right? So inside the cell, there is continuous production and degradation of the RNAs. And when we talk about gene regulation, what we really want to measure are these individual events, right? The rate of transcription, the rate of production, the rate of decay. But then what we can really observe from transcriptomic profiling, like Rna-Seq and at the older times microarray, is the abundance of the mRNA, the mature RNA. So basically, we just have a snapshot of one of the products in this process. And that makes it difficult to study these steps. So these are the things that we're really interested in, the rate of RNA production and decay and processing. But what we really can measure is only the abundance of the RNA. And when we talk about a process and its rate and the dynamics, we always think about time point measurements in order to understand the dynamics of a process. But I think the question that we'd like to answer is whether it is possible to look at a snapshot of the cell, for example, using Rna-Seq, and still be able to measure the rates of these dynamic processes, the rate of RNA production, processing, and decay. There's a recent preprint by Adam Sippel's lab from Cold Spring Harbor, where I took this very beautiful figure from, that explains this concept very well. So we can liken the life cycle of RNA to basically a bathtub where water is pouring in, and then there is a drain with a certain diameter where water is pouring out. And there is a balance between how much water goes in and how much water goes out. And because of this balance, the level of the water inside the bathtub remains the same. Now, this is RNA production, the water that goes in. This is RNA degradation, the water that goes out. And this level of water that we have in the bathtub is the steady state RNA. The interesting thing is that even with a snapshot of this picture, if we look at, for example, the steady state level of RNA, and if we knew some other kind of information, for example, if we knew how much RNA is being produced, so how much water goes into the bathtub, we could still measure how much water goes out of the bathtub. So basically, we could measure the diameter of this drain. And this is exactly what we are interested in. This diameter is really the RNA degradation rate. And the idea is that if we somehow know the steady state RNA abundance, which is easy to get, the Rna-Seq gives us that, right? And also somehow we know the rate of RNA production, then we would be able to measure the rate of RNA degradation, basically as the ratio of RNA degradation, RNA production versus steady state RNA abundance. But how can we measure this RNA production? So in the old times when we were looking at Rna-Seq data, we would see this pattern, right? So these are reads from Rna-Seq. There is a concentration of reads at the exonic regions of a gene. But then what we would observe was also these few reads, relatively few reads, like less density, but actually because the introns are long, a lot of reads. So we would see these reads that map to the intronic regions. And there was a time we would just ignore these because we thought that these could be, for example, genomic contamination. But then we realized, and also a lot of other people showed, that actually these intronic reads come from unspliced premature RNA inside the cell. So when we do Rna-Seq of the RNA content of a cell, it's not just the mature and processed RNA that we capture from the cytoplasm, but we also have unprocessed RNA from the nucleus that we capture. And these manifest themselves as these intronic reads. So basically, what happens is that in this process, we don't have just one measurement. In reality, we have two measurements from two different points of this process. And that is, I think, the key to understanding the rates of those individual processes. So I think this figure kind of shows this concept a little bit better. Consider a condition where we have very fast transcription of a particular RNA, but also we have very fast degradation. So in other words, low stability of a particular RNA. What we see is that we have a lot of reads that map to the intronic regions. So a lot of reads from unspliced RNA, but fewer reads from mature RNA. And that's because the mature RNA gets degraded quickly. On the other hand, in a different condition where we have slower transcription, we see fewer reads from pre-mRNA because pre-mRNA is closer to the transcription. So slow transcription means fewer pre-mRNAs. But then if we have really high stability, we would see a larger concentration of reads at the splice junctions. So this is the idea. The idea is that by looking at how exonic reads change from one condition to another condition and how intronic reads change from one condition to another condition, we can basically infer whether we have an increase or decrease in transcription versus an increase or decrease in degradation. Obviously, really quantifying these rates from these spliced and unspliced RNA abundance is challenging, and that is where one of my really great students, Rashid, comes into the picture. So back in 2017, we published this paper and this tool that we call RemRems, where basically we model how the exonic abundance or change in exonic abundance is expected to follow with the change of intronic abundance if there was absolutely no change in RNA degradation. So we go back to some very basic assumptions about the biochemistry of the different kinetic rates of RNA. And we try to say, if there was no change in degradation, then what would be the expected relationship between exonic and intronic reads? This would be the expected relationship. Now, any deviation from this expected relationship would indicate a change in degradation rate, so a change in splicing. And this is what we can actually measure from Rna-Seq data. So we have two conditions. We look at how exonic reads change. We look at how intronic reads change. We would predict how the exonic reads would have been different if there was no post-transcriptional modification. And then we look at the actual data. We measure this deviation, and then that tells us whether we have post-transcriptional regulation or not. So at that time, we applied this method to different tissues from the human body, normal tissues, and also we studied it at the context of neurodegenerative disorders, which is its own interesting story, but I won't go into the details of it. But what I'd like to mention a little bit more today, talk a little bit more about, is the uncertainty in these measurements that we have from Rna-Seq data. So here we rely on intronic and exonic reads basically to see how the unspliced and spliced RNA has changed across conditions. But we know that Rna-Seq data are noisy, and the reads that we get from, for example, exonic reads or intronic reads are actually noisy measurements from the actual abundance of mature RNA and premature RNA. And now we're taking these noisy measurements and we're taking their ratios, and then we take their logarithm, and then we compare these logarithms. So that amplifies the noise really severely, which means that even if we really had no change in RNA degradation, when we look at all these transformations, because of this noise, we actually see a wide distribution of observed ratios in intronic and exonic reads. So there is a big uncertainty when we measure these ratios from count data. And that is what one of my other superstar students, Gabriel, has been working on, to see if we can actually quantify this uncertainty by properly modeling the properties of sequencing read counts. So basically, the idea is to model the read counts as a probabilistic noisy measurement of some underlying latent variable, which is the true abundance of spliced reads and unspliced reads, and then model these latent variables as functions of the transcription rate and Rna stability, and Rna stability itself would then be a function of the condition that we are interested in. So for example, let's say that we are comparing the disease state of a tissue to the healthy state. So mRNA stability would be modeled as a function of this condition. And then can we actually come up with the distribution of all these latent variables based on the observed kind of noisy exonic and intronic read counts. And also recently, another student, Wenmin, has joined in the fund trying to model these latent variables with a probabilistic based solution. And what we have right now is a method called DIFRAC, so differential ratio analysis in count data, which can actually give us estimates of RNA stability along with confidence intervals and along with statistical metrics that tell us whether the stability of an RNA is significantly different across two tissues. So Gabriel has been using this method to study the changes in RNA stability across different cancer types using, for example, TCGA transcriptomics data. So the idea is to apply DIFRAC to the tumor and normal samples across different tissues and see what are the genes whose stability changes from healthy tissue to tumor at the stability level. And I think it's fascinating how many genes are regulated in cancer at the stability level. So this is what we get. The majority of genes have a different stability in tumor compared to normal in at least one of the different cancer types that we study. And there's a big amount of cancer-specific pattern, but there are also patterns that kind of repeat themselves across cancers. And we can look at the pathways and biological processes that are regulated in this way using RNA stability in cancer, and some really important pathways pop up. So, for example, we have oxidative phosphorylation, whose genes seem to be downregulated at the stability level across multiple cancers. So basically, the genes that are involved in oxidative phosphorylation are destabilized, their mRNAs are destabilized in tumor. And you know that oxidative phosphorylation is kind of a big aspect, an important aspect of cancer. We have this Warburg effect where the tumor cells switch from an oxidative situation to rapid metabolism through glycolysis. So we have downregulation of oxidative phosphorylation. And these analyses suggest that a lot of this happens at the level of RNA stability. And then we have other processes that are actually more stable in tumor in terms of their RNA half-life. So for example, we have genes involved in epithelial mesenchymal transition, another very important process in cancer, that seem to be upregulated across multiple cancer types at the level of mRNA stability. So I think this is really fascinating, and it's an aspect of cancer that we haven't really looked at, and that's how genes and their expression changes in terms of RNA stability. So we have all these measurements. We now can say whether a particular RNA is upregulated or downregulated at the RNA stability level in cancer by looking at transcriptomics data. But is there a way to actually figure out how these changes happen and what are the molecular factors that drive these RNA stability changes? So one of the classes of factors that we have looked at are the Rna binding proteins. And these are really interesting proteins. So the human genome, depending on who you ask, has anywhere between 200 to 2,000 RNA binding proteins. And in one of the studies that I participated in my postdoc, we looked at the sequence specificity of 100 of these RNA binding proteins in the human genome. And what we saw is that each RNA binding protein recognizes more or less a different and unique sequence within the human genome. So they recognize these sequences within the RNA itself and bind to the RNA, and then that is how they affect the mRNA in terms of its processing and in terms of its life cycle. So it's pretty much like transcription factors, the way that transcription factors recognize specific sequences in the promoter, bind, and regulate transcription. These RNA binding proteins form these regulatory networks through recognizing specific sequences in the RNA itself, binding to them, and regulating the RNA. So now, the idea is knowing, is something like this, knowing what kind of genes and what genes are bound by each specific RNA binding protein, and therefore knowing the regulatory network of each RNA binding protein. And also knowing the behavior of these RNAs in cancer, which RNAs are upregulated, which RNAs are downregulated at the stability level. Can we combine this information in order to figure out how the activity of RNA binding proteins change from cancer, from normal to tumor tissue, for example, in cancer? So basically identify the regulators, the factors that drive these changes in RNA stability. Look at the behavior of the targets of an RNA binding protein and infer how that RNA binding protein itself behaves in cancer based on how its targets behave in cancer. So there are a lot of approaches that people have established through the years for looking at gene expression changes, combining it with gene regulatory networks to figure out the activity of regulatory factors. One of the simple approaches that I really like and I think is really powerful is by modeling, for example, the abundance or the stability of an mRNA as a function of the effect of different RNA binding proteins on that mRNA. So this effect can be positive when an RNA binding protein binds to mRNA and increases its stability, or it can be negative when it binds and decreases the stability of the mRNA. So a function of this effect times the abundance or activity of that RNA binding protein in the particular tissue that we are studying. The higher the activity of an RNA binding protein, the larger effect it has on its target mRNAs, and the lower the activity, the lower effect it has. So this gives us this kind of a linear system. So it's very simple. It's basically a regression-based system where we know the abundance and stability of mRNAs. We know the effect of RNA binding proteins on each RNA based on previous research. We know the regulatory networks. We know whether an RNA binding protein destabilizes or stabilizes RNA. And all of that we can combine in order to infer the abundance and activity of the RNA binding protein using a regression approach. So one of the first cancer types that we looked at using this method was clear cell renal cell carcinoma. It's the most abundant type of kidney cancer. And the reason we chose this cancer was that there are a lot of genes whose expression changes between tumor and normal. And what we were interested was to see whether these changes could have happened at the level of RNA stability, right? So we basically used the methods that I described to measure the stability of RNA. We combined it with these regulatory networks that I mentioned that were constructed from previous work. So we knew which RNA binding protein binds to which mRNA. We had these regulatory networks. And then we could infer the activity of RNA binding proteins using this method. And it showed several RNA binding proteins that seem to have prominent effect on the stability of RNA in kidney cancer. For example, there was this PCBP2 RNA binding protein whose activity seemed to be really increased in tumor compared to normal, because when we looked at its targets, basically we saw that the targets of this PCBP2 were kind of uniformly upregulated at the stability level in kidney cancer. And then other RNA binding proteins like MBNL2, whose activity seemed to be increased, and ESRP2, another RNA binding protein that seemed to have decreased activity in tumor. So these are inferences from our model. Do they actually hold true when we look at these proteins? So the first thing that we did, I'm going to highlight one of these RNA binding proteins. The first thing that we did was to look at the Rna-Seq data from these RNA binding proteins directly and see how the RNA that encodes these proteins changes from tumor to normal. And surprisingly, for example, for this protein PCBP2, we saw that the RNA almost doesn't change between tumor and normal. So our model says that PCBP2 is upregulated very significantly in tumor, but then Rna-Seq data says that the RNA that encodes this protein isn't really different between tumor and normal in terms of its abundance. So what is going on? And we decided to look at the abundance of the PCBP2 protein itself. So instead of looking at this RNA, let's use antibodies, immunohistochemistry to look at the abundance of PCBP2 in tumor samples and compare it to the normal samples. This is a work that was done in collaboration with Yasser Yazal-Hosseini's lab at McGill. And we saw this surprising huge change in the abundance of the PCBP2 protein itself when we go from normal tissue to the tumor tissue, which is consistent with what our model predicts. And we were able to further validate that this protein, PCBP2, actually has an important role in regulating the transcriptome of cancer cells. So when we look at knockdown data, so if we knock down PCBP2 in cancer cell lines, do Rna-Seq to measure the changes in expression as a result of knockdown, and then compare it to transcriptomics data from tumor, we see that the same genes that are upregulated in tumor versus normal, the same genes get downregulated when we knock down PCBP2. And also the reverse can also be seen. Genes that are downregulated in tumor versus normal, they are the ones that are upregulated when we knock down PCBP2. So there is at least a partial reversal of the tumor transcriptome when we knock down PCBP2. And it's interesting that a lot of these genes that are downregulated by PCBP2 knockdown are actually involved in cell cycle. So when we look at their functions, we see there is a huge enrichment of cell cycle related genes. And Yasser, my collaborator, had this fantastic postdoc in his lab who actually went and knocked down PCB2 in these cells and looked at how cell cycle changes. And he confirmed that cell cycle actually gets downregulated. So basically, we have an arrest in cell cycle when we knock down PCB2, which I think is really interesting. So it's an example that highlights how modeling RNA stability and then combining with network information can reveal factors that can't be really identified in any other way. If we were just to look directly at the expression of PCB2 RNA in cancer, we would not be able to identify this relationship. So that was the story about kidney cancer. We published it a while ago, but then now remember that Gabrielle is looking pan-cancer at changes in RNA stability. And what we'd like to know is whether there are RNA binding proteins that are involved in regulation of RNA stability across different cancer types. So a similar analysis with this pan-cancer landscape of RNA stability gives us a picture like this. So there are a lot of RNA binding proteins whose targets are significantly upregulated or downregulated across different tissues. But then this analysis also highlights these two proteins, RBFOX1 and RBFOX3, that seem to be downregulated across multiple cancers. So when we look at their targets are destabilized in tumor compared to normal across different cancer types. And that's really interesting that we have this factor that whose regulon is affected across different cancers. So if we look at, for example, HITS click data, that tells us where these RNA binding proteins bind. We see that they bind a lot to 3' UTR of many different genes. And the genes that have a binding site in their 3' UTR for these RNA binding proteins, they seem to be downregulated at the stability level across different cancers. And there is this subset of genes that are kind of uniformly downregulated in many different cancer types. And we were able to validate that actually these genes are really under the regulation of RBFOX1 protein. So if we look at Rna-Seq data from knockdown of RBFOX1, we see that these genes are downregulated when we inhibit the expression of RBFOX1. This is a gene set enrichment analysis, shows downregulation of the majority of these genes. And then on the other hand, if we re-express RBFOX1 in cells that naturally do not express this protein, so basically if we introduce ectopic expression of RBFOX1, then we can also rescue the expression of these genes. So we have these genes that are bound by RBFOX1, they are downregulated across multiple cancers. They are downregulated when we knock down RBFOX1, they are upregulated when we rescue their expression. So all of these suggest that RBFOX1 is really an important factor that modulates the expression of these genes. And we are now looking into the functions of these genes and what is their effect and their role in cancer biology. But a quick glance at the data tells us that there is this core set of genes that seem to be really involved in calcium signaling pathway. And downregulation of the RBFOX proteins destabilizes these genes in the cancer cell, causes their downregulation. And we know that calcium signaling pathways are really important pathways in regulation of cell cycle and cell proliferation and many other cellular pathways. So maybe that is how these proteins are involved in determining the behavior of the cancers. So RNA binding proteins are one class of factors that determine mRNA stability. There's also this other class of factors, the microRNAs, that I think are very well studied in the context of cancer. So the human genome contains thousands of microRNAs. And if you search the name of any microRNA on PubMed, probably the first hits that you see some connection between microRNAs and cancer. And the way that these microRNAs works is, again, they recognize specific sequences within the target mRNA. They bind to these mRNAs and they cause degradation of the RNA along with translational inhibition. So what we'd like to know was whether some of this change in mRNA stability that we see across different cancers is mediated by these microRNAs. So basically come up with a global picture of which microRNAs change RNA stability between tumor and normal in every cancer type. And this is the picture that we get. many different microRNAs affecting RNA stability in different cancer types. But then we have something like this microRNA, miR29, whose targets seem to be upregulated across multiple cancers. And remember that microRNAs inhibit RNA expression. They cause RNA degradation. So when the targets of this microRNA are more stable in cancer, it means that the microRNA itself is probably less active in the tumor cells. And miR29 is actually, when we look at its expression itself, is downregulated in cancer, so that makes sense. But what we tried to do was to actually see whether we can rescue some of these changes by reintroducing miR29 into cancer cells. So again, in collaboration with Yasser Riaz Al-Husseini's lab at McGill, the work of Puria shows that, okay, if we reintroduce miR29 in some cancer cell lines, so for example, 786O cells, which are models for kidney cancer, unsurprisingly, we see that the targets of miR29 are downregulated when we reintroduce miR29 into these cells. But more importantly, we see that the mRNAs whose stability is downregulated as a result of reintroducing miR29 are the same mRNAs that are upregulated in kidney cancer compared to the normal tissue. So this corroborates this hypothesis that a good portion of these stability changes in these cancers, for example, can be caused by downregulation of miR29. And I'm not showing the genes here, but these genes, when we look at their functions, we see that they actually have very important functions. A lot of them are, again, involved in cell cycle. A lot of them are involved in cell proliferation. So I think with that, I'd like to kind of wrap up these stories. I hope that I could convince you in this presentation, basically, that RNA stability is really important. As I mentioned, not many people are looking at RNA stability and basically these post-transcriptional modifications in cancer. But by being able to measure the rate of RNA decay and then applying our method for RNA decay measurements to pan-cancer data, we can see that there are a lot of genes whose expression changes at the stability level, and it's the rate of decay that determines which genes are active in cancer. And then we can combine this information with networks that we know from factors such as Rna binding proteins and microRNAs to model the function of these factors. And basically that tells us that we have these master regulators, such as the RB FOX proteins and miR29, that are really important across different cancers, and they recognize key cancer-associated processes. So they regulate the genes that are really involved in the biology of cancer. And then the other thing that I'd like to highlight here is really the power of public data, public transcriptomics data. So you might have noticed that basically the core of our discoveries comes from publicly available data that people have put out there, from large-scale consortia such as TCGA to small-scale studies that have looked at the effect of individual factors. And with appropriate computational models, we can really learn a lot from these data that were not really the key purpose these data were developed. And then we can go into the lab and validate these hypotheses, which I think is really interesting. So with that, I'd like to acknowledge the lab members. I think that I mentioned the people who were doing really key work in this aspect from my lab, but also from Dr. Riaz Al-Husseini's lab at McGill, and also our collaborators at UCSF that have helped us with validating these different computational methods by actually measuring RNA decay, showing that our estimates are really a good match to RNA decay rates, and also other collaborators who have helped us with different measurements. So thank you very much. And I think that if there are any questions, I will be very glad to answer those questions.
Thank you, Dr. Najafabadi, for this exciting and nice talk. We have a few questions. Maybe you can see in your Q&A tab.
Yes, so
I would read the first question. To capture the intronic parts of the pre-RNA, do you need to carry out total RNA experiment, or can some signals also be of and in poly-A enriched libraries, which is most commonly used in the protocol? And I would also like to extend this question by asking you that in terms of alignment, do you need any specific tools to align to get the better better quantification of exons and introns?
Excellent question, really. So total RNA is obviously preferred because when we look at the total RNA from many different tissues, what we really see is 50% of reads or maybe 60% mapping to the exons, but then another 40 to 50% mapping to the intronic reads. So that is really optimal for this kind of inference. But we have been looking at poly-A enriched libraries as well. So I think actually TCGA data is poly-A enriched in many different cases. And we see at least 20 to 30% intronic reads in poly-A enriched libraries. And I think the reason is that, okay, the RNA processing is not really stepwise process, but really everything happens at the same time. So we might have some unspliced RNA that is already polyadenylated. So we can capture these reads, and that has been enough actually to measure RNA stability in our applications, I think. And in terms of alignment, so we have a process where basically we add the intronic reads back to the annotations when we want to measure the RNA abundance. So we create these GTF files where the intronic regions are also in the GTF. And then with the basic genome alignment that we have, we can measure the abundance of these intronic reads. But some of the people in my lab, so for example, Rashid, and also a really good postdoc that I have, Rick, have been looking at how we can do this with better tools. So for example, can we use tools such as Salmon or Callisto and introduce the pre-mRNA as one of the transcripts in the annotation, and then look at these alignment-free approaches to measure their abundance. So there are a lot of different ways of doing this, and I think that we're just starting to learn what tool is optimal. But it is true that we need to modify the process a little bit.
Another question is that it is known that some exons and introns can work as enhancers. Have you seen any difference in terms of degradability?
Yes.
I guess. Yeah.
Yes, very interesting questions. Honestly, we haven't looked at it. And I think it would be super exciting to do that. So do the introns or exons that have different functions other than just being part of the unspliced RNA, do they also have differences in terms of degradation? I think it's a very interesting question to look at.
Okay. This is the last question because we are already a little bit over time. We know the deficiency of some of the RBPs result in alternative splicing. How can we differentiate the unspliced intronic reads from the reads which align to the intronic relation in the spliced mRNA? And would there be a normal isolate for the intronic sequencing coming from pre-mRNA in both conditions?
So this is really a key point. And we try to kind of make sure that as much as possible, we remove the effect of alternative splicing on our measurements by, for example, looking only at exons that are exons no matter which transcript we look at. So we call this the constitutive exons. So these are exons that are not alternatively spliced in or out across different transcripts. And we try to do the same with intronic reads. So based on available annotation, we look at introns that are introns no matter what kind of transcript we have. So basically, we try to remove this effect as much as possible by concentrating on these genomic regions that do not show evidence of alternative splicing. And the second part of the question, I think, was that would there be a normalization for the intronic sequences coming from pre-mRNAs in both conditions? So we separately normalize for intronic and exonic reads in our process. And the idea is that from sample to sample, we might have different capture efficiencies between spliced and unspliced RNA. So we need to normalize them differently. I hope that this answers the question. But I can definitely go into more details later.
I would like to read this last question.
Sure.
You need to be modeling expert to look at the mRNA decay rate, or are the models that you have developed now also available as a tool, or for example on
Oh,
GitHub, etc., and we can download them directly and use them?
of course. So we have put these tools available for download on GitHub. And I tried to put a link in my slides, but also if there's another way to also share these links, because I don't know if everyone has access to these slides or not, I'll be happy to share them. But we have tried to essentially create these tools that you can just download, and then there are simple steps that can be followed with protocols that we have also included in the documentation to measure RNA decay rate. And if anyone wants to use these methods and has questions, we will certainly be happy to help.
Okay, thank you very much for this nice and interesting talk. And
Thank you very
we
much.
would like to also inform all the attendees that if you have more questions, you can contact Dr. Najafabadi directly and chat with him more. And if you are more interested in this kind of research, talk to him about this. And now I would thank you again. And
Thank you.
now we would like to